So Peter's going to get here. He'll be back here in a second to start his talk. But we have somebody who has recently arrived, Yen from Singapore, from A-Star Singapore. He's actually in Peter's lab. He's an expert in multi-scale modeling, what Peter's going to talk about, and he'll be here for the rest of the week. So I know some of you have interest in doing coarse graining in association with atomistic models in one simulation, and he is. Talk to him if you're interested in that. Okay. He's right. Can you raise your hand, Ian? Right there. All right. I'll hand this off to Peter.
Thank you very much. Okay, so yeah, thank you very much for letting me contribute to this. I think it's a very nice workshop, and hopefully we can give you some useful information on the multiscale approaches, which is really the focus of our lab. So before I go into the details of that, I just wanted to mention, we're based in Singapore, as already mentioned, work as part of A-Star, which is really it's a government institution, but it's really an umbrella set of multiple institutions. So there's a whole range of different kinds of science going on in Singapore. Some of it is biomedical, but there's also kind of engineering and electronics and so on. But we're very much on the biomedical side, based in Biopolis, which is this kind of biomedical research hub, combination of really academic and pharma, biotech, and so on. So it's quite a nice kind of ecosystem. And really what I just wanted to highlight is there are multiple opportunities for both undergrads and potentially for PhDs as well. So we have a range of PhD studentships, ones which can either be done fully in Singapore and as part of a kind of a joint PhD between A-Star and a local university. But we also have these PhD studentships, which are kind of a program which is really supposed to support collaboration. So it's kind of typically something like two years in a host university in another country, and then 2 years in Singapore. But then the other thing for any potential master's students who are interested in coming, we have these pre-graduate internships, and they typically can be as short as 2 months, but they can go up to 6 months. And they're, and actually I heard recently there are rumors that they may even go up to 9 months now. But they're fully paid, and I think it's a nice opportunity for a bit of travel and to experience something different. So if you have any interest in any of those, or if you know anyone else that might be, then please do either give me a shout, or Igor, who just spoke. And then, of course, you met Fergus last week, who is not here, but I'm sure you're still in contact with him. And then Jan, Jan is somewhere here. Jan's at the back here, and he's going to stand up and wave so that everyone knows who he is. There we go. There's Jan. So he's very happy to talk to you guys as well. Okay, over to the science. So I always like to start when I'm talking about simulations with the classic example, which I'm sure you're very familiar, you're probably not, of my dog. So my pet dog is a bulldog, and I like to think about this in terms of as in kind of a metaphor for thinking about structures and dynamics. So we know about biology. We're typically used to thinking about biology in terms of textbooks, kind of schematics. But of course, we like to go beyond that and start to think about 3D structures, as we're doing here. And if you just focus on the 3D structure of my dog, my pet dog Bella, just based on this particular structure, you might think that it's a very static, very boring, sleepy structure. But as it turns out, on very rare occasions, she may become quite dynamic. So in this case, she's jumping around on the bed, which I like to think of as the free energy landscape. She's interacting with possibly a ligand here. She's doing something functional here. And this is really the idea why we want to use simulations and modeling to go from just static structures and schematics to really understand the mechanisms of these things. And yeah, so this is obviously the analogous example where we're actually really thinking about molecules here. And I won't go into too much detail now because I'm going to talk about this a bit later. But just to reiterate that this is a case of an antibody, an IgM antibody, and we can go from a schematic to an integrated model of this thing, and then we can also simulate dynamics. So I know that you've already had an introduction to MD, so I'm not going to spend too much time on this slide. Yeah, I'm not going to spend too much time here, but I just wanted to remind you that obviously you've already been introduced to the idea of a force field, which is really the underlying kind of the key to getting accurate results in your simulations. And so we have, a combination of bonded and non-bonded terms. And of course, we are very lucky these days, at least compared to when I started my PhD a few years ago, a few decades ago, in that the force fields are really incredibly accurate now. So, and you already heard about things like D.E. Shaw and Anton, who have these incredible computers that allow you to run very, very long simulations and watch your proteins fold up kind of spontaneously. So although that example is perhaps becoming slightly defunct these days with things like AlphaFold, I think it's worth reiterating that, structure isn't everything. So of course, the way in which you get to those structures is also very important. And so you have to think about the dynamics and the mechanisms, and that's where simulations come in. But even then, even with these very accurate force fields, that's not always sufficient to give you the answers that you want, or for some reason this isn't working. Sorry, bear with me. Okay, it's advancing on my computer, but not on the screen. I may need technical support here. Maybe I just cancel it. Oh, there we go. Okay, here we go. So yeah, although we have these beautiful, fantastic, accurate force fields, not all of us have access to these very, very impressive specialized computers like Anton. And that can be a problem because, if you want to just simulate your protein for a few nanoseconds and look at the wiggling and the jiggling of the atoms, then that's fine. But, really, we really want to think about the functional motions and of course, we all have our own particular projects and our own particular questions in mind. But oftentimes, the time scale is really, really important. And so typically, we may want to go out to microseconds or even beyond that, even up to the millisecond time scale. So think about things like large-scale conformational changes and ligand binding, even protein folding and assembly of multiple proteins or proteins and DNA and all the rest of it. And similarly, as computational methods advance, we also know that there's been kind of an explosion in structural biology-based approaches as well over the last few years. And again, these are things which tend to sample on the timescales of milliseconds or seconds or way beyond that. And if we want to, parallel between our computations and our experiments, then we need to really think about those sorts of time and length scales. And of course, the problem with, running these simulations on a typical desktop computer or maybe even a computer that has, say, 20 cores or even 100 cores, who knows? It's really tough to be able to sample those sorts of timescales. So this is 10 years old, so it's a little bit out of date, but I don't think it's probably still fair, depending on the size of your system, that if you're just working on a desktop computer running these simulations, then you may need a year to run a microsecond timescale simulation. So you're not really going to get very far there. And of course, if you want to go out to seconds, then you're going to have to wait for millions of years, and for most of us, that's not an option, unfortunately. So what's the alternative? What else can we do here? So one alternative is to take these kinds of multiscale approaches to go out to these coarse grain methodologies. So at a very basic level, and I'm sure some of you have heard of coarse graining, really what we're doing is we're trying to find ways to simplify our system whilst still retaining the key the key features of that system that still describe it accurately. And one way that we can do that is we can take groups of atoms together in our system and we can group them into larger particles, as you can see here. And that automatically obviously simplifies the system and speeds up calculations just in terms of reducing the number of particles in the system. So that's already a good thing. But typically we also then simplify the kinds of potentials, the descriptions, especially of the non-bonding interactions that are going on in the system. And that again gives you typically an order of magnitude speed up. So those two things together means that we can start to run these simulations in a coarse grain resolution at a much faster. We can generate useful data at a much faster rate, and maybe you're looking at, rather than waiting for a month for your simulations, you're looking at maybe just a day. So that's the good side. The bad side is that obviously, a lot of care is needed with these kinds of simplified models. They are ultimately very simplified models. So the accuracy of the predictions are really only as good as how good your coarse grain model is and how carefully parameterized it is. And then on top of that, there are things like kinetics, which, kinetics really is something that you don't necessarily expect to get accurate kind of estimates of from your coarse grain models, because the force field is much more simplified and much smoother. Of course, there are ways to make sure that things are working properly. So one thing is to ideally what you would have is you would have an all-atom model, and ideally all-atom simulations as well, that you can use to kind of parameterize your coarse grain model, or at least to validate it and make sure that what you're getting from your coarse grain resolution is close to your atomistic resolution. And then on top of that, we have this advantage now that we can do all these kinds of integrative modeling approaches, which I know many of you are interested in. And with this recent explosion in structural biology techniques, ranging from very high resolution methods and X-ray and NMR, going to the medium resolution, although this is also becoming high resolution now with cryo-EM. And then things like hydrogen deuterium exchange will give you information on dynamics, atomic force microscopy, and then omics data as well, which allows you to, for example, describe accurately the glycans on your protein or the lipids in your lipid bilayer. And all of those things you can incorporate into your coarse grain models and try to increase the accuracy of those simulations. And then finally, one thing to say is that, typically these days, quite routinely, we can actually switch between kind of back and forth between these coarse grain and atomistic resolutions. And so that typically gives you a kind of a best of both worlds approach. So it means that you can run your coarse grain simulations for a long time, and then you can switch back to atomistic resolution and maybe refine your models, for example. So before I go into the nitty-gritty of this stuff, I just wanted to give you one example to show you the power of these sorts of approaches. So in this case, we recently, this is actually work from Ferdus, who was here last week. And so he was looking at some antibody drugs, and which are already used in cancers, in treating cancers. And so they used basically to target these HER2 tyrosine kinase receptors, which we know dimerize and can lead to and result in cell growth and division. But the problem is that the gene is typically amplified in many cancers, and when that happens, what we want to be able to do is use these particular drugs to target those and slow down this dimerization and stop the cell growth. So these particular drugs, trastuzumab and pertuzumab, which I always struggle to pronounce, are in the IgG form of the antibody, which is what most of these kind of monoclonal antibody drugs is the most common isoform that typically is used. But then what we asked was, well, we know that we can also have these IgM isoforms of antibodies, which automatically have more antigen binding sites. So you would think that that's going to be something that can be advantageous. Maybe you can increase the avidity and the binding affinity of these kinds of drugs and maybe be more effective. So really, that was the question we asked. We said, well, how about what if we think about the IgM form of these two particular drugs? So this is jumping ahead of myself, because actually we did the calculations first to make the predictions, but I just wanted to give you the final result from the experimental lab, from the wet lab. And basically what they showed was that indeed pertuzumab IgM is indeed better at inhibiting cell proliferation than its IgG form. But that's not true of the other drug, of trastuzumab. So that's kind of weird. So why is that? What's the reason for that? And the way that we tackled this was to use a multiscale approach. Why do we need to do this? Well, because IgM is already a pretty huge complex. So it's something like 8 to 10,000 residues in total, and that leads to several million atoms. And so obviously running long timescale atomistic simulations is going to be pretty tough for this size of system. So first of all, we built our integrated models for these, initially for the IgG form of these drugs. I won't go into too much detail here, but we used a combination of homology modeling based on X-ray and NMR, and various nice structures were available to be able to do this. And then what we can do is we can run atomistic simulations of these protomers, and we can watch them flop about dynamically. And then what we can do is we can do exactly the same thing using a coarse grain model of these structures and run simulations of those as well. And really the aim of this is to validate our initial coarse grain models. And so one example of validation here, all I'm doing here is to measure the distances between either the FAB antigen binding domains, or between the end of the antibody and those FAB domains. So two different kind of dynamic descriptors of the way in which these things are moving around. And what we can do is, so up here on the top, we have the atomistic dynamics, and below we have the coarse grain dynamics. And what we can see is that we do a pretty good job here of reproducing those overall dynamics. So that gives us confidence that what we've derived with our coarse grain model is accurate. So once we've done that, we can start to move on to the big things. So here, now I'm going to talk about our full-length IgM multimers, hexamers or pentamers of both of these drugs. And so we can run, first of all, we can run simulations of this IgM hexamer, and we can show that there's an interesting protrusion here in the C-terminal region. Again, I won't go into too much detail, but it's something that we could then validate by comparing back to cryo-AFM data, which showed very similar things, of course, at a lower resolution. But then we can also think about the dynamics. So if we run long time scale coarse grain simulations of these hexamers or pentamers, then we can then calculate the root mean squared fluctuations, which is just a kind of formally related to crystallographic B factors, and gives you an idea of how dynamic these things are. And then we can calculate an average density of this thing from our simulation. And then again, we can look through the literature, look for any useful structural biology data. And in this case, we have some nice negative stain EM, which really matches up very nicely. And it shows that the center of these kinds of complexes are very, very stable, whereas the outside are much more dynamic. And that's pretty much what we see from our simulations as well. So now we have some confidence that what we're doing is reasonable. So now finally we come to the core question. How do we think about explaining why these things are binding more or less efficiently to their targets in cancerous cells? So what we know is that there are some nice crystal structures available for both of these drugs in their monomeric form bound to HER2, and we know that they bind to different places. And then, so first of all, we think about pertuzumab. So this is the one which in its IgM form, in its hexameric form, in the wet lab binds very, very nicely and inhibits these cancer cells. And what we can see is from our simulations after modeling all these HER2s bound to this antibody is that there's no kind of significant overlap here, and everything's very stable, and the complex looks like it's perfectly viable in the cell. Now, if we look at the one which isn't working so well, then what we can see here is that now when we bind these ligands onto their targets, there's significant overlap between the antibodies and the antigen. And so what basically that says is that only one of these HER2 epitopes is able to bind to the antibody. So it doesn't actually make any sense to then design a IgM antibody which is not going to be able to bind more than one ligand anyway. And so and if we look at our simulations and make some predictions based on the space between these binding sites, then we can set what we can say fairly confidently, in only 0.1% of all our microsecond timescale simulations are kind of more than one ligand going to be able to bind to these pentamers and hexamers. So that nicely explains the wet lab data. And as I said, we did the computations first and then got them to do the wet lab experiments just to confirm our prediction. So I think that's hopefully a nice example of how you can use a combination of atomistic and coarse grain simulations to kind of almost drive these kinds of experiments. And this is something that we hope going forward in the future can be something that's going to be useful for other antibody therapeutics. So I did promise that I would go into the nitty-gritty a little bit. I'm not going to go into too much detail here because I think probably that's something that we may do in the following year. But just very briefly, so the first thing to say is that there are a whole range of different sorts of coarse graining approaches and force fields and so on. But really the hugely popular one, which I'm sure many of you heard of, is Martini. And this is really, you know, almost ubiquitous these days. It's gone through various iterations over the years. We also contributed ourselves from our own lab to contributing some of the models and parameters. At the moment, we're in a kind of a transition period. So we know version 2 of Martini is very good. Version 3 was released in a kind of beta version not so long ago. It's being used more and more frequently, but from the experience in our lab is that we're still not fully confident that it's that it's accurate for all systems. So I would suggest some care there. What we typically do these days, and what I would suggest you do if you're interested in using Martini, is to perhaps use both version 2 and version 3 and see which results which simulations give you better results. In terms of what's inside Martini, although there are various iterations, it hasn't really changed that much over the years. It's been refined, but you know, the fundamental building blocks of this force field have remained the same. So typically what you have is you have something like 4 sets of heavy atoms which get grouped together into these larger particles. So I have some examples here of water, or an ion surrounded by water, or a lipid, where we see that some of these atoms on the head group are grouped together into 1 or 2 particles, for example, and similarly for the acyl tails. And then you have a series of bond and angle potentials, very similarly to your atomistic models, but those bond and angle potentials tend to be much softer. So again, this kind of softens the energy landscape, allows you to sample much faster timescales. One key thing to bear in mind that, you know, all of these are simplifications, and in particular the electrostatics are very, very simple. You know, you're not using things like E-world summation. Typically, you're using kind of relatively short-range electrostatics, and we do know that long-range electrostatics are important for governing a whole range of biomolecular processes. So again, it's something just to bear in mind when you're running these kinds of calculations. So this is, I've got a couple of slides actually from work that we did a few years ago when we were developing our own kind of Martini-compatible protein force field. I wouldn't recommend you use this now, but it's just really to give you a taste of the kinds of processes that you would use to develop these sorts of models. So as I said, we're grouping sets of atoms, you know, typically 4 sets of atoms into larger particles. So you can see some amino acids here, which are represented by 2 or 3 kind of blobs. And then we have various ways to try to validate this. So one thing that we can do is we can actually do free energy calculations. So what we can do is we can calculate the free energy associated with transferring, in this case, amino acids from a non-polar phase to a polar phase. And that's something that you can do experimentally, and you can also do computationally. And so that gives you really a good way to really validate that the energetics of these sorts of systems are accurate. And we can see here, for the most part, that we do a reasonable job in correlating simulations and experiments. But then we can start to look at things like transferring these things across lipid bilayers and calculating the free energy for that. And again, that's something that we can correlate with experiment or with atomistic simulations as well. And then we can look at larger peptides. And so there are a series of peptides, well-known peptides. It doesn't really matter what they are, but there are a series of well-known peptides which are nicely, you know, kind of, I guess, studied for a long time as model systems in terms of using things like spectroscopy to work out where they're partitioning within lipid bilayers. And so we can confirm that, you know, these things are orienting either along the interface or they're inserting into the membrane. So just one recent example, just to show you the principle of, you know, deriving new building blocks, because although Martini is very good and we have, you know, some nice parameters for proteins and amino acids and lipids, sometimes you may want to look at a new kind of molecule which isn't necessarily kind of part of the force field as yet. So what we do in this case, this is an example of glycosaminoglycans, which we know are important in a whole range of kind of cellular recognition processes, and they're also important for things like viral infections, for example. So what we can do is we can choose look at our glycosaminoglycans, look at these chains made up of these kind of charged sugar units, and what we do is we can kind of manually actually just kind of, you know, based kind of almost on chemical intuition, we can group together these sets of atoms into particles. We can choose them ourselves, kind of make an initial guess. We don't necessarily have to stick on those options. We can always refine them later on. And then what we can do is we can join these things together. We can choose a set of kind of generic bond and angle potentials with generic force constants, and we can watch these things jiggle around. So this is an atomistic representation of a simulation of this glycosaminoglycan, and this is our first pass representation of our coarse-grained version of this. And what we can see is actually it looks a little bit rigid. It's not really reproducing the kind of helicity that we see in the atomistic representation. But then we, as I said, we can quite quickly run these simulations, so then we can start to iterate this thing. So we can start to match up the bonds and angle potentials to better match up with the atomistic simulations. And so our final model here, hopefully you can see, is doing a pretty good job of kind of, jiggling and wiggling in the same way as the atomistic chain. And then finally, in terms of validation, again, just to remind you again, we can also do this in a slightly more rigorous way in terms of the non-bonded interactions. And so what we can do is we can calculate, again, the free energies of partitioning into a polar phase like water and a non-polar phase like benzene, for example, or cyclohexane. And we can just make sure that for these longer peptides, or even for the individual constituents, we can make sure that the free energies are matching up with the experimental ones. And again, if they're not, then we can go back to the drawing board, or not necessarily the drawing board, but we can go back one step and we can change the particle type of individual groups on these glycosaminoglycan chains. And this is something, although this work is not published, it's something that we did recently for N-glycans, for example, so a very similar sort of process. So it sounds like a lot of work. Actually, once you kind of get to grips with coarse-grained approaches, then actually you can run these calculations very quickly. What I'd also recommend is that if you're interested in doing this kind of thing, then you go back to the original Martini papers, and certainly there are a few which really almost provide you with a kind of a step-by-step guide on how to do this. So going a bit further and starting to think about just beyond these simplified polymers or small peptides, we of course often are interested in running simulations of these of large proteins, or even protein complexes. So here, typically the simplified coarse-grained potentials for describing bonds and angles of dihedrals don't always do a great job of reproducing the underlying protein dynamics. If that's the case, then there is another opportunity, and that involves using this kind of fairly simple elastic network approach. So really all you're doing is you're choosing a cutoff between sets of particles or atoms, and just introducing elastic restraints to kind of stop this protein from falling apart. And you can see this is really an example here. This is a simplified outer membrane protein called OMP. And what we can do here is we can look at the RMSFs, the root mean square fluctuations of this protein during the simulation in both atomistic and coarse-grained resolution. And we can show that in this case, the overall pattern of fluctuations match up really rather nicely. So you can see that these lower regions here, the lower magnitude regions are the beta strands, and then the very high regions are the loops at the top here, which are moving around a lot. If you don't get good agreement, then you can just go back and you can play around with either the cutoff of this elastic network to restrain atoms that are further apart from one another, or increase the force constant. So there are a range of ways that you can kind of calibrate these models. So that's the kind of a brief introduction to the nuts and bolts of these things. So I just wanted to show you a few examples of why this is useful. So this is just one example here. So this is this protein that I just showed you before. And what we're doing here is we just throw it into a box surrounded by randomly placed lipid and water molecules. And what we can show is that over the timescale of hundreds of nanoseconds, the lipid bilayer gradually forms spontaneously, and this protein inserts itself spontaneously into the lipid bilayer. And again, just to reiterate, this simulation took something like, I guess, a few hours, whereas if you were doing this atomistically, then you're probably looking at a few months. Similar kind of example here. This is in the case of a couple of peptides which are known to dimerize, and we can again see the formation of the lipid bilayer and the dimerization of this thing. And that, without showing you the details, this matches up actually pretty nicely with available NMR data. It doesn't have to be proteins, of course. There are models now available for DNA, RNA as well, and then more recently some work that we did looking at nanotubes and the way potentially some nanotubes which we're trying to develop that can kind of carry anti-cancer peptides into cells, and this is something that was recently published. Sorry, I'm going to have to do the same thing again, I think. So going on from that, just one very quick slide to emphasize, I guess, ultimately really what one of the main aims of using these coarse grained approaches, of course, is to really be able to tackle very large systems that we're definitely really going to struggle to do atomistically. And so without going into too much detail, so one example up here was looking at an antimicrobial peptide and trying to get to the bottom of its mechanism. So our experimental collaborators were using wet lab experiments using vesicles and watching these things kind of penetrate these vesicles and leading to leakage. And so indeed, with these kind of coarse-grained approaches, we can pretty much reproduce the experiment in near atomic detail. And we can watch as all of these hundreds of peptides absorb onto the surface of a lipid vesicle and then lead to this pore formation. And then one other example, which again really wouldn't be possible using atomistic approaches, is to think about these cyanobacterial thylakoid membranes. So the reason we were looking at this was because you can actually overexpress certain enzymes in these cyanobacteria, which are able to synthesize alkanes. So obviously this is of interest from a kind of a fuel development or fuel perspective, I guess. But the problem when you start to overexpress these enzymes is that it seems that the alkanes somehow disrupt the electron transport chain within the membranes of these things. And so they wanted to understand why that is to try to stop that from happening ultimately. So what we can do is we can simulate these very, very large membranes, thousands of lipids, 20 different lipid types, and then we can add in, alkanes, in this case heptadecane, and we can just see what happens as you start to increase the concentration of these alkanes. And what you can see is that when you get to a critical concentration, these things start to intercalate between the leaflets of the lipid bilayer, and at some point even start to cause things like curvature and even large-scale phase transitions, which of course is not going to be good for these curved membranes within the thylakoids. So that's a brief overview, kind of, I guess, the earlier stuff to do with coarse-grained approaches. And then in the next few slides, really what I want to do is give you a taste of what we're doing in our lab, and in particular with a focus on host pathogen interactions and how multiscale simulation approaches can help with this kind of stuff. So I'm going to focus on two main things. One of these is going to be on viruses. So we tend to do a lot of simulations of viruses, particularly dengue virus, of course, coronavirus as well, as I guess everyone is interested in these days. So dengue virus is something which I suspect many are familiar with, but it's a virus which is transmitted by mosquitoes, and really, we'd be interested in trying to develop antivirals or antibodies, vaccines, and so on to be able to stop this, the kind of the global burden of this. And then the other area that we're interested in looking at is bacteria. In particular, a major focus of ours is on gram-negative bacteria, which cause unfortunately, a lot of dangerous infections these days, particularly because of the growing crisis of antimicrobial resistance. We particularly focus on these lipid molecules on the outside of the gram-negative bacteria, these quite unusual kind of long hydrophobic but also highly charged lipids called LPS, lipopolysaccharide. And we look at this both from the perspective of the bacteria, but also from the perspective of the immune system, because we know that there are specialized receptors called toll-like receptors, which are able to bind to these LPS molecules, and that kind of is used as a signal to tell the immune system to respond to these kinds of infections. That sounds like a good thing, but actually quite often when you get an overwhelming bacterial infection, actually what you get is that these LPS molecules overstimulate these particular receptors, and that can lead to kind of whole body inflammation and death. So this problem of sepsis is really a global problem, which if we could develop new therapeutics for that, then obviously that would help a great deal as well. So I'm going to, first of all, I'm going to talk about this toll-like receptor system that I mentioned just then, and at the end, I'm going to talk about some of our work on dengue virus. So the toll-like receptor system, what is it? So it's quite a complex multi-component system made up of multiple sets of proteins, and in part, this is probably because these LPS molecules are very hydrophobic. So it's difficult to really get hold of them to be able to recognize them. And so you have specialized proteins that can extract these lipids from the bacterial membranes, then pass them along to this, an accessory protein called CD14, which then passes it along to this, the final toll-like receptor. And if this toll-like receptor is able to recognize that these lipids are dangerous, that they're coming from pathogenic bacteria, then they're able to switch on the immune system and start the immune response. So yes, and this process of recognition involves dimerization of these toll-like receptors, and that's really the start of the signaling cascade. So this is kind of the story, well, I say so far, but this was the story from a few years ago where we were using just purely atomistic simulations to look at individual sets of these proteins. And really the key take-home message here was that we could use atomistic simulations to simulate just part of this toll-like receptor complex bound to its co-receptor protein called MD2 in the presence of a range of different LPS analogs. And what we could show is that in the case of certain antagonistic lipids, so lipids that aren't going to switch on the immune system, the complex is very, very stable, very unstable. And so actually what we see is that there's a key residue, a key phenylalanine in this MD2 co-receptor protein, which when you have antagonist bound, it will kind of flip from inside to outside, and that will destabilize this complex and stop it from activating. On the other hand, if you have a very kind of agonistic stimulatory ligand, such as an LPS molecule from E. coli, for example, then this phenylalanine is very stable and the complex is very stable, and that will allow signaling inside the cell. And so that allowed us to make a range of predictions and help us to understand why certain novel ligands are able to switch on or switch off this toll-like receptor inflammatory system. So we had a reasonably good example, really reasonably good handle on this kind of so-called molecular switch and how this was working for just the individual complex. But really what we wanted to be able to do was to go beyond this, to go beyond schematics, as I say here, and really try to understand the entire system using these multiscale approaches. So how do we do this? Well, what we need to do is we need to kind of systematically parameterize each of these coarse-grained components, using a coarse-grained approach. So this is the LPS molecule, the key region of the LPS, which we know binds to these toll-like receptors. What we can see is that we have these multiple acyl chains here, we have some sugar head groups, and then we have these charged phosphates as well. And so we can use a similar sort of approach. We can assign different groups of atoms into larger particles, and then we can start to tune our bonds and angles. And at the same time, we can run atomistic simulations and make sure that the overlap of the bond potentials or the bond distributions and the angle distributions and the dihedrals are matching up nicely. And so we do this in an iterative way. It sounds like a lot of work, but actually really only takes a week or two to be able to do this because these coarse-grained simulations are so quick. So, and then how do we validate this? Well, we can do various things. So one thing we can do is use these kind of potential and mean force calculations. So we can slowly pull our lipid out of a lipid bilayer here, and we can gradually calculate the free energy associated with extracting this lipid. And this is exactly what we're showing here in the graph form. So this is the free energy, and you can see as we slowly pull this out, then we can see that there's a big energy cost associated with extracting this lipid. And we can do this in both atomistic resolution and coarse-grained resolution. And we can confirm that we're getting good match up there. And we can do various other things. So we can check the structural and dynamic properties and compare these to available data from fluorescent labeling and X-ray diffraction, and make sure that all these kinds of properties are at least within error, within the limits of our experiments. So I've already mentioned about this molecular switch, and of course, so we're confident now that our lipids are doing the right thing, but how about the proteins? And because of the importance of this molecular switch, we need to make sure that this is doing the right kind of thing. So one thing that we know from our atomistic simulations is that If you run a simulation in the absence of a ligand, what we see is that this protein actually collapses. This co-receptor protein that normally binds the lipid, it collapses, and this phenylalanine is kind of exits the binding site. And that's exactly what we see from our coarse-grained protein after we've carefully tuned the elastic network model here. And then the second thing that we can do is that we can just we can drop a lipid onto the surface of this protein and just watch and see if it binds to the binding site. And hopefully you can see here as these lipid tails slowly worm their way inside the binding site, and finally, they actually end up pretty much taking up the position that we see actually in the overlaid crystal structure here. So we've got to the point where now we know that this molecular switch is behaving sensibly at the coarse-grained resolution. And we do similar things for this CD14 co-receptor as well. So now we start to build the models, construct them all. So here we have our TLR4 bound to MD2. We have the transmembrane domain, insert it into a lipid bilayer. Do the same thing for CD14. It's GPI anchored, but again, we're binding this within a lipid bilayer. And then what we can do is, I've talked about the free energy calculations that we did for our LPS molecule, but we can do exactly the same thing for all the components of this pathway. So we can do it for extracting from the lipid bilayer, but also from CD14, from MD2, and from TLR4 as well. And to cut a long story short, what we can basically show is that we can calculate the free energies to go from the bacterial outer membrane all the way to its terminal receptor complex, TLR4. And we can show actually quite nicely that there's a kind of an overall free energy funnel which favors the extraction of this lipid and is binding to this terminal receptor complex. So that's nice from a kind of a quantitative and confirmatory point of view, but we can also start to do some fun stuff here. So what we're doing here now is we throw together our receptor and our co-receptor, and we can just watch as these things start to spontaneously interact with one another. And at some point, you'll see that these things come together into a potentially kind of active binding mode. And indeed, if we run these simulations for long enough, we can actually watch this lipid get transferred from this CD14 protein to this TLR4 MD2 complex. So we can really start to see the detailed mechanisms of how these things are actually working. Okay, so one final thing to say in the context of this particular system. So it sounds like this is something completely different, but it's not at all. So we've been working with a collaborator for several years now who are interested in the thrombin kind of clotting pathway. And what we know is that during the clotting pathway at a wound site, parts of this thrombin protein get cleaved off. And for many years, it was assumed that these small or large peptides that are thrown away are just kind of lost and recycled. But as it turns out, they seem to have interesting kind of anti-inflammatory effects. And so for some of these larger peptides, what we could show is that in silico, in vitro, and in vivo, these peptides are actually able to aggregate LPS and almost scavenge this LPS. And so what we think is going on here is that this is really kind of dampening the inflammatory response at these wound sites to prevent this kind of over-inflammatory potential. But then also for some of these smaller peptides, in the last couple of years, what we were able to also show is that it seems as though some of these peptides really have direct receptor binding activities as well. So they seem to be multifunctional. So on the one hand, we can use coarse-grained simulations to show that these peptides are able to bind to these LPS aggregates and stop them from being passed along to the TLR4 chain. But then in some cases, with one particular peptide in particular, derived from thrombin, we can show that it actually binds directly to the CD14 binding site, so the LPS binding site in CD14. And so that's acting as a competitive inhibitor to prevent, to prevent this overinflammation from happening. And we can show it in vivo as well. Our collaborators were able to perform some various kinds of experiments to confirm that indeed there is this competitive inhibition going on. And also, we managed to get them to do some cross-linking experiments with mass spec to show that indeed even the actual position, pretty much the exact position where we were predicting this peptide was binding, was indeed the place that we predicted. And so this is quite promising as a potential anti-inflammatory therapeutic, and we're in the process now of trying to modify the peptide, let's say, to maximize its potential as a drug in the future. Okay, so I think in the last few minutes, I just want to tell you briefly about our work on dengue virus, and again, emphasize the use of being able to use these multiscale approaches where you can switch between atomistic and coarse-grained resolutions to study these huge systems. So very briefly, just an introduction to dengue. So we know there are hundreds of millions of cases around the world every year. It's a big problem obviously in Southeast Asia and certainly in Singapore. And one thing that we know is that actually the first infection of dengue with one particular serotype doesn't necessarily lead to too many serious symptoms in most cases. But if you get infected with a different serotype, then this can lead to something called antibody-dependent enhancement. So actually your antibodies are able to recognize the virus from a different serotype, but not necessarily actually be able to neutralize it. And so that can actually lead to a worse infection. So of course, it's a huge burden, at least economically, around the world, and obviously makes a lot of people miserable. So we want to use these integrated modeling and multi-scale approaches to try to get some insights into the mechanism of the life cycle of these viruses. So this is an envelope virus containing a lipid bilayer and envelope proteins on the surface, and then the nuclear capsid inside the virus. We're lucky enough to have some very nice cryo-EM structures of these viruses. So you can see the arrangement of these envelope proteins on the surface of the virus. But unfortunately, of course, we can't see where the lipid bilayer is. And because it's an envelope virus, we know the lipid is really important to understanding the life cycle of the virus. So that was really our first aim, to try to come up with an accurate model for the entire dengue virus. So how do we do this? Well, actually, this is work that Jan largely did over several years. And really, so it's a big effort incorporating all sorts of different kinds of data, including glycomics data, also lipidomics. So we can tune the contents of this lipid vesicle according to experimental data. But then really the key thing here is, again, although we're using the Martini force field here, we want to make sure that our protein dynamics are behaving sensibly. And so again, what we can do is we can do simulations of things like just small fragments of this virus. So in this case, maybe something like a dimer of these envelope proteins in atomistic and coarse-grained resolution. And then what we can do is we can compare the dynamics between the two sets. So we can look at things like root mean squared deviation, so look at the structural drift of the protein, also things like residue contacts, as I'm showing in a very small image here. And ultimately, if we tune out our elastic network model carefully, then we can get to the bottom of these things, and we can get a reasonably good model. So that's obviously a good start. So how do we validate this? So one thing that we can do is we can run our coarse-grained simulations of our virus over relatively long timescales, over a few microseconds. And what we can do is actually we can calculate the experimental density map, or rather we can calculate a theoretical density map based on our average simulations, our average simulation trajectories, and then we can directly overlay this with the experimental density map from cryo-EM. And so what I'm showing here is that at 0 nanoseconds, we can see actually if you overlay those two, then obviously there's not a lot of correlation because we're initially starting with this very spherical vesicle, lipid vesicle. But then as we run our simulations, we can start to see this correlation goes up. And by the end, we have excellent correlation here, something like somewhere around 0.8 between the theoretical and experimental density maps. And so that really provides us with a means to not only come up with a reliable viral model of this envelope protein, of this envelope virus, but we can also actually use this approach as a general approach to try to help out our experimental colleagues in terms of refining these cryo-EM maps. So now we have some confidence, we can start to do some more interesting things, I think. So one thing that we tried to do was we also wanted to try to look at the spiky version of this virus. So typically the virus, when it's floating around, is very smooth and spherical. But when it starts to become infective inside endosomes, then it adopts this spiky state. And of course, I'm sure everyone has heard of spikes these days because of COVID. So we know that the spikes are the ones that start to do the damage. And so when this transition occurs, these spikes contain these fusion peptides, which allow fusion between the virus and the endosomal membranes. So this was quite a complex problem because, unfortunately, we don't have any high-resolution maps for these very unstable, spiky versions of the virus, but we do have some low-resolution cryo-EM, and so we can combine taking a combination of X-ray and NMR structures and some simulation tricks, which I don't have time to talk about now, but Jan, I'm sure, will be happy to tell you. And we can also do things like, for certain regions of the protein which we can't see in our cryo-EM maps, we can at least refer to things like hydrogen deuterium exchange, which gives you information on the dynamics of certain regions of these things. So that allows us to at least make some decent guesses on how dynamic some of the missing loops are, for example. And then what we can do then is to generate a series of different sizes of viruses containing different numbers of lipids. And we can look for the one which is most stable. So once we have that, we have our starting model for this spiky infective virus. Before I talk about that, I just wanted to mention that we can also do these kind of toy models. So in this case, our toy model is just one or a few of these spike proteins represented in coarse grain resolution interacting with an endosomal membrane. And this is already quite interesting. So what you can see here is that as you start to add these spikes into the viral membrane, you can see that as it interacts with the endosomal membrane, it starts to induce this curvature. So this is already quite promising and starts to give some insight into how these kind of fusogenic processes are happening. But yeah, of course, the fun stuff, the thing that we really wanted to do was look at the whole virus when it's binding to one of these endosomal membranes. And we get some quite nice results here. So we can see that this thing spontaneously fuses with this endosomal membrane. And if you run it for long enough, you start to see these kind of long hypothesized hemifused states as this mammalian endosomal membrane and the viral membrane start to fuse. And we can see this is quite cholesterol dependent as well, as an aside, which we know experimentally is true as well. So I hope that gives you an example of some of the things that we can do using this combination of atomistic simulation, coarse grain simulations, and integrative modeling as well. Again, just to reiterate, we always do this in very, very close parallel with experiments. So something else that we've been doing focusing on the dengue virus is thinking about its overall dynamics. So what we know is that I think for most people would think of a virus as a very kind of static structure, but actually these things are quite dynamic, and they can become compact or expand in response to things like fever. And this probably correlates to the fact that, at least in the case of dengue, it's transferred from the mosquito, which is colder, to the human, which is warmer, obviously. So you seem to have this expansion as you increase the temperature. And so we can combine things like cryo-EM to drive our models in the compact and expanded states to look at this. And we can also then start to look at the reversibility of this process. And working with experimentalists, we can switch back and forth between atomistic resolution. And the motivation for this was that what we knew from fluorescence is that in the presence of certain ions, this kind of the expansion is reversible. And using atomistic simulations, we can show why this is. From a therapeutic perspective, one important thing that has emerged from the last few years in terms of this very dynamic dengue virus is that actually, in response to certain point mutations, this virus can become either more or less can tend to be more or less kind of morphologically dynamic, I guess. And so what we could do is, in this case, what we can do is we can switch our coarse grain resolution virus back to an atomistic resolution, but just for this dimer of envelope proteins, and we can then use atomistic simulations in the presence of different point mutations to predict why certain mutations are going to lead to a more or less dynamic or more or less stable kind of viral surface. And so that's actually really important from a therapeutic perspective, because it starts to tell you that, when you're developing things like antibodies or vaccines, then you really want to have a vaccine that's able to drive the immune system to recognize these multiple different morphologies of the virus to make sure that you're able to neutralize all of them. And then, so this is, I think, just the final example of something that we've been doing with dengue virus, and it's something else which is therapeutically pretty relevant. So I've already mentioned to you the fact that there are multiple serotypes of dengue, and this can be something that can lead to serious infection if you get multiple infections by different serotypes. But so, of course, we would like to understand why this is. I think it's something that's been long, for a long time, is people have been aware that it's a phenomenon, but it's not really clear why that is. And in terms of this seems to relate to the maturation of the virus. So it turns out actually that dengue is not very good at kind of maturing. So as it's secreted from the cell, what it needs to do is it needs to shed this so-called PR protein to allow these spikes, to allow the envelope proteins to become exposed and then for the virus to become infectious. But actually, many dengue serotypes don't seem to do this very well. And so that seems counterintuitive, but actually if you think about the fact that this seems to offer an advantage in terms of this antibody-dependent enhancement, then that actually makes evolutionary sense. So working with our collaborators who are doing cryo-EM, they were able to solve a series of structures of this virus when bound to some of these antibodies and during the process of maturation. Unfortunately, the resolution of these cryo-EM maps were very, very low. But of course, now we can take advantage of this and we can combine these cryo-EM maps with our coarse grain models for the virus. And what we can do, for example, is we can use these kind of steered MD approaches or kind of biased approaches to drive our viral model between these different states, so between the different levels of maturation. And we can do this. What we can watch is as this happens, we can actually see that gradually during this process of maturation, these PR proteins get knocked off in the presence of antibodies. And if we look at these systems in the presence of the antibody as well, then what we can see is we can actually see the reason for this is that these antibodies start to clash with one another during this process and knock each other off. So that seems to be the reason why these antibodies are favoring this maturation process and making these viruses more infectious. So that's obviously a start. It doesn't, it starts to give us some insight into the mechanism of this process of antibody-dependent enhancement. Of course, the next step would ideally be to target some of the regions on the virus that are being recognized by the antibodies and try to block that. So obviously, that's something that we're thinking about in the future. So this is just my last couple of slides. Hopefully I'm not talking for too long, but this is kind of an aside. So this is something which I'm not sure if people will have already talked about to you during the workshop. But this is something called kind of solvent mapping approaches. So this is something where we know that there are often on the surfaces of proteins there are kind of hidden or cryptic pockets which are not necessarily exposed in the crystal structures, but which potentially can be druggable. So one thing that we can do is we can use simulations in the presence of things like benzene molecules, and what we can actually look for pockets that are opened up by these kinds of benzene molecules. So this is a way to try to look for novel druggable pockets, basically. So this is a method which has been used for several years now, but we wanted to be able to do it for viral proteins. The problem with these kinds of viral proteins, of course, is that they sit in a lipid bilayer. And if you're throwing in a load of benzene, then where's the hydrophobic benzene going to end up? It's going to end up in the hydrophobic lipid bilayer. So we developed an approach whereby, by adding a series of additional potentials, we can stop these benzene molecules from entering the lipid bilayer, and that allows us to then explore the surface of the protein. So after running lots of these simulations, what did we find? Well, one thing we found was that we found a pocket called the beta OG pocket. So it's actually a pocket which is already established. So it's a pocket which was discovered a few years ago on the surface of the dengue envelope protein. And it was observed because fortuitously a precipitant molecule was found to bind to this site and open it up. And indeed, from our benzene mapping simulations, we can see that this pocket is also spontaneously opened up here. So that's a nice validation of the approach. But then what we can also then do is obviously then to start to look for other kinds of pockets. And we did indeed observe one interesting pocket, which we termed the alpha pocket. Why is it interesting? So the reason that it's interesting is because it seems to be the site of a series of highly conserved charge residues, including some histidines, which have been long hypothesized to be important in terms of acting as a pH switch. So when this virus is inside the endosome, the pH drops. That triggers a conformational change in this envelope protein and allows it to open up and expose these spikes for fusion. So we wondered whether we could explore this. And so really, this is an example of, although I've mostly been talking about multiscale approaches in terms of simplifying things, actually in this case, what we're doing is we're actually using constant pH simulations here. So this is going a bit more detailed. So what we can do is we can actually perform simulations of this envelope protein where we can allow the ionization state of these charge residues inside this alpha pocket that we observed. We can allow those charge states to change. And as we gradually lower the pH, indeed, what we can see is that the charge state of this particular conserved cluster starts to change. And as that happens, we see this spontaneous opening of this protein domain, which is the domain that needs to start to open up for this protein to become spiky. So yeah, I just wanted to mention this at the end, just to say that multiscale doesn't necessarily have to mean simplifying things. So I think I've probably come to the end here. So just to summarize, so I hope I've just at least given you a taste of what these kinds of coarse grain and multiscale approaches can do. We can look at really massive systems. We can look at complex systems and the way in which different proteins and other sorts of biomolecules can assemble with one another. The key thing with all of this is, of course, that we have to do it very carefully. We need to validate our models either using atomistic simulations or ideally by working closely with experimentalists as well. So I will probably finish there and just thank the people that did the work, really. So Jan is here, and Fergus, obviously, you met last week, and really they've done a lot of this work, and then the whole team as well. And of course, thank you to the funding, who we wouldn't be able to do any of this without. And I will finish by leaving it on this slide again. So, once again, just give us a shout if you have any queries or if this is of any interest to you or any of your colleagues. So, yeah, and I'm happy to take questions. Okay.
Okay. Yeah, thank you for that very impressive presentation. I have more like a functional question regarding the antibody-dependent enhancement, which you just presented for dengue. And since it has been known for dengue virus for a long time, and so far, at least to my knowledge, we have not seen anything, despite the expectation that we would or that we should, we have not seen anything for coronavirus. Do you know why that is? Or is there anything known about this?
Yeah, honestly, I don't know. So I mean, we have started to work or we have been working for a couple of years on coronavirus, particularly the spike. The antibody-dependent enhancement, I don't know. I mean, I don't know if maybe it's too early. I'm not really sure. It's something that I'm not personally particularly familiar with at least on the biological side of things. So
right.
Yeah, I wish I could comment, but
yeah, no
unfortunately
problem.
I can't.
Thanks. No problem.
Thank you, Peter, for the question. For the wonderful presentation. With regards to the ADE, there's this notion of original antigenic sin, right? And T cells are also thought to be involved. Have you thought about looking at the role of T cells with regards to this? Thank you.
Yeah, no, I mean, yeah, it's a very good point. I mean, I suspect that the ADE, I mean, I think we're looking at just one very small aspect of it, and probably there are indeed multiple aspects to it. There's probably other antigens and epitopes involved, and indeed, indeed T cells as well. It's something that, honestly, it's something that I've so far not really looked at in too much detail because, as I said, I think we'd like to work closely with experimentalists, and so far we've not been able to convince them to go down that route. But yeah, I'd absolutely love to, and I guess in the past we have looked a little bit at some other aspects of T cell, receptors on T cells and so on, more from the point of view of things like activation of the transmembrane domains and the ITAMs and all the rest of it. But yeah, I think it's such a complex problem. I suspect we're decades away from really truly understanding it properly. But yeah, I'd love to.
May I ask something?
Yeah, sure.
Very impressive. I really would like to learn more about that. Okay, so there are some practical aspects to it. Number one is the preparation of your systems, okay? And the other one is the timescales. Just now you mentioned some time that it might take months to get it done. And yes, because if I have to see a transformation of one atom also to another one, or get an insight, I need to do it on larger timescales. So how do we contain it within the graduate time period? For example, a PhD student doesn't have like so many years maybe, and he has got a lot of work to show. This could be just one part of his entire thesis. And then again, we have to show that this is proven in the labs. How do we correlate them? The timescales? How do we manage them so that we can get some good results, validated results to be proven? And the preparation of the system is definitely an issue. So how do we get around that problem?
Yeah, it's a good question. I mean, I think it's a multi-pronged problem. I mean, one trick really is to obviously be careful with what kind of system, depending on what question you want to ask, what biological question you want to ask, be careful about what sort of system you choose. I mean, one advantage, of course, with doing these simulations of the virus, for example, is that we have multiple repeating units of this protein. So although indeed, yeah, if we wanted to validate our coarse grain model of the virus by running a full atomistic simulation of the virus, it just doesn't make sense, right? But what you can do is you can choose maybe a small repeating unit, icosahedral unit from your viral system, from your virus. So in this case, this is what we did with dengue, and we were just simulating a dimer, an atomistic dimer of these two proteins. So we can run that quite quickly. That takes a few weeks to get some reasonable dynamics that we can then calibrate our coarse grain model with. It's not always as easy as that, but that's one way to do it. The alternative, which is, I guess, well, talking about coronavirus, so we've also been doing various coarse-grained studies of the spike protein. Obviously, that's a huge system. We can do atomistic simulations of that, but it takes a very long time. But then that's where we turn to the literature, and that's why I always tell my students, literature search is the most important thing right at the start of the project. And what we can do is we can, for example, by going after we can look at things like there's data on, from cryo-EM and cryo-ET, for example, showing how this spike protein is kind of flopping around in either just a single membrane or in the context of the entire virus. So this is beautiful data that it really is useful for us. So we can run our coarse grain simulations. We can check the angles that it's kind of rotating with respect to that lipid bilayer, and then we can compare back to the experiments. And if it doesn't match up, then we can run another coarse grain simulation where we kind of stiffen up the potentials. And when we get something that's behaving properly, then that gives us a good starting point. So I think the key is to, one, talk to as many people as possible who know about the biology of it and the structural biology, and two, literature search. And then I think as long as you're strategic about how you go about it, then I think you have a good chance of being able to do that in a reasonable time frame.
So I just wonder what are the advantages and disadvantages relative to like enhanced sampling methods such as accelerated MD, for example.
So you mean using that in the context of the coarse grain approaches, or?
It seems like you're trying to both simplify large systems, also be able to and sampling should be able to access larger time. So how would you compare it?
Yeah, I mean, it's a good question. I mean, so certainly we do use enhanced sampling approaches. I think it really depends on the problem. I mean, I talked at the end about these benzene mapping approaches, which or in a sense is kind of an enhanced sampling approach. And of course, we use things like replica exchange and so on. When we're doing that, we're tending to look for maybe, things like druggable pockets or maybe slightly more subtle changes. With the coarse grain approaches, you're not necessarily going to be able to, there are definitely some deficiencies. So large scale conformational changes within a single domain is something that you're not going to be able to get using this kind of coarse grain approach unless you add in some extra information and you drive the conformation of that protein from one state to another. So I think it really depends on the problem. Although I would say that what we like to do is to be able to switch between the different resolutions. So, for example, we can use the coarse grain approach, say, to assemble a membrane protein target within a lipid bilayer. We can switch to atomistic resolution and then use an enhanced sampling approach to look for a subtle conformational change or where a drug might bind, for example. So I think that really I would not necessarily say one is better than the other, but I think it's part of the toolbox basically.
Hey, Peter, wonderful talk. So for the small molecules, like the recent Martini paper, they were using small molecule. Is there an automated way to generate the small molecule parameters yet? And or is it something you're working on? Where do we stand?
Yeah, I mean, So I mean, there are a range of approaches available. I know I'm aware of several approaches where there are papers already where people have kind of semi-automated it, either using using kind of a approach where kind of iteratively just matching up different kinds of angles and dihedrals and all the rest of it, or of course more recently using machine learning approaches to derive potentials on the basis of the atomistic sampling, and that's something that we're certainly doing as well. So yeah, I mean, there's a range of approaches, but I would also, again, I know I keep repeating it, but I think that there's no substitute for the human eye as well. And I work closely with someone who was using these kind of automated approaches to develop a model for LPS, and although technically it was exactly correct, it was almost kind of over-tuned, over-parameterized compared to the atomistic model. And so actually our one where we incorporated a little bit of kind of expert knowledge, I guess you would say, allowed us to get correct behavior of our lipid protein binding and things like that. So certainly there are automated approaches available, but yeah, I think always check carefully, basically.
And then about the peptide, in that coarse grain model for the parameters we develop for peptide, can the peptide become unstructured, restructured, or it maintains only a single structure?
Exactly. So this is another thing where the secondary structure is typically enforced. I mean, so there are things like there have been some papers which have developed kind of these Go-like models, for example, on top of the Martini force field. And so I don't know how well that would work for small peptides, but I guess certainly for larger proteins, then that might be a useful approach if you really want to look at these conformational changes. We tend to take a more conservative approach. We may use atomistic simulations to characterize 2 or 3 folded and unfolded states and simulate those individually and try to make sense of those from the coarse grain, the individual coarse grain simulations.
And then finally, a technical question: as your system gets bigger and bigger and bigger, and so you run it on GPU right now, right?
So we mostly use GROMAC, so then it's mixed CPU, GPU. Yeah.
For the coarse grain part, when you get bigger, so do you have to connect between multiple GPU? I'm just thinking when should we be there when you can simulate the whole cell using coarse grain, and what will be the technical bottleneck for there at that time?
Yeah, I don't know. I mean, of course, there have been some amazing studies in the last few years looking at massive systems. Romy Amaro looking at the entire coronavirus. I think we're certainly getting there. And obviously, there have also been simulations of very kind of crowded cellular environments. So, yeah, I think it's oftentimes it's a matter of just having access to the biggest and the best resources, frankly. But I'm always a little bit wary about how useful the data that comes out of those things are. When often if you can, I think you can get more out if you have a focused question and you want to get an answer for that rather than just simulating as big as possible and then looking for something interesting at the end of it. So, but yeah, I don't know. 20 years time, I'm sure we'll be able to simulate a cell.
Okay, thanks.